Okay, so welcome everyone. It's a pleasure to have with us today Miguel Lurgi, who is presently lecturer in computational ecology in Swansea University, but he has a long history. He graduated from Simón Bolívar University in Caracas, Venezuela, in 2003 on computer science or computer engineering, and then he did two masters on artificial intelligence and robotics in Sussex and Edinburgh, the UK. And then he moved to Barcelona to do a PhD on theoretical ecology in the Center for Ecological Research and Forestry Applications in the Autonomous University of Barcelona. Then, okay, since then he has moved in several positions, for example, in Microsoft Research in Cambridge, UK, in Trinity College, Dublin. He was also in the University of Adelaide in Australia, in the Experimental Theoretical Ecology Station in southern France. And since 2019, he's senior lecturer at Swansea, the UK. And now, recently, just a couple of weeks ago, he was positively evaluated for a Ramón and Cajal position. So he's looking for some place to develop the Ramón and Cajal research project. The research topics of Miguel are a variety of them, but essentially centered on ecological networks, network dynamics, epidemiology, microbial ecology, global change, meta-communities, so quantitative descriptions of ecological processes. And today the talk will be titled towards a mechanistic understanding of the assembly and disassembly of ecological networks. So Miguel, the floor is yours.
Cool. Thanks very much, Emilio. That was a very kind introduction. I appreciate. Yeah, as Emilio said, I try to do a little bit of computational ecology, try to mix like theory and some data analysis. And I am not really a physicist, and I don't pretend to be. I'm far from it. So I apologize in advance for the low technical aspects of this talk. It's gonna be probably for most of the audience is gonna be relatively easy to follow. But yeah, I just wanted to share what I have been doing over the last I guess like 7 years or so, and yeah, I just hope that you like it. So I'll start with giving a little bit of background for those of you who are not ecologists or not familiar with the sort of theoretical food web approaches that I will be talking about. Then I'm going to talk a little bit about how we look at assembly mechanisms in my lab, and also how to use that knowledge from assembly to try to develop a more mechanistic and predictive understanding of the effects that these stressors will have on complex communities. And then I'll present like a couple of examples of how I have used models like this to improve conservation strategies, basically. So my main interest in terms of community assembly is to try to understand how what seem to be like random collection of species, like this picture, can actually be represented in a more like sort of structured way. Like, for example, this example of a simplified version of the North Atlantic food web. Even though this object seems to be like even more complex than the one on the left, the interesting thing about this is that, there are things, well, you almost must be familiar with networks and stuff. So the interesting thing about these objects is that we can quantify things about them, right? We can count stuff. So the sort of my whole motivation, and that of many ecologists for a few decades already, it's to try to understand what are the mechanisms that make these things to be organized in this way. And it all started with ecologists like Hutchinson and MacArthur and Margalef even, and Elton, back in the 60s, where they were trying to basically answer, like simple questions such as why are there so many species? How can they all live together? What are the consequences of these interactions for fluctuations and the stability of the system? And in general, how to better understand community structure and assembly. So back in the day, the whole thing was, let's say, a little bit phenomenological. Until like around the 70s, Robert May, who actually was a physicist, he came up with this more quantitative approach to understand how collections of nodes in general in a network, but applied to ecosystems in particular, whether there are some limits to how a system like this can grow. And he came up with this famous equation in theoretical ecology, which is some sort of rule, I guess, based on random matrix theory, that says that in order for a system to be like feasible or stable, the interaction strength between species, which is I here, multiplied by the square root of number of species times connectivity has to be smaller than 1. And he realized that, in general, in natural systems, this, it's very hard to have a very small system that will comply with this strategy or with this rule, even though, there are some examples in laboratory conditions, like, for example, this work by Fox and McBrady Steele, who had actually proved that this rule is true in real ecosystems. But even so, he, May was always wondering about why we see so much complexity, right? And he basically posed the challenge back then to identify the devious strategies that natural system uses to increase its species and connectivity capacity without sacrificing stability and persistence. And ever since then, basically the whole point or the whole goal of many ecologists, like including myself, is to find what are these devious strategies, right? And I tried to do this in order to be able to then create predictive frameworks. Once we know what are the mechanisms or these devious strategies, then how predictive frameworks grounded on these mechanisms can help us better understand how systems like this will react to different kind of perturbations happening either alone or at the same time, like warming, extinctions, invasions, overexploitation. And this is the sort of things that I currently think about. And I try to tackle these problems using a sort of mixed empirical and theoretical approach, in which on the sort of theoretical modeling side of things, I use different sort of approaches like, network dynamics based on differential equations, more qualitative type of network structural analysis. And more recently, also adding a little bit of like meta-community and individual-based approaches to this. The individual-based approach will come, will be evident why this is important in one of the studies I'll present. And I try to, relate this also to the analysis of empirical data in many different systems. Like, for example, this network is a representation of the microbial communities living inside marine sponges. Then I have looked at the geographical variability of intertidal communities across the Southeast Pacific. We have also looked at the scaling of network structure across different biogeographical regions in Europe. And the whole point is to try to analyze all of these systems and come up with generalities like this. This is an example of a degree distribution for the sponge microbiome network, and we can see that the sort of they follow what is expected from this kind of network, some sort of like fat tail distribution. So in terms of community assembly, I have looked at like different things, but here today I'm only going to focus on the drivers of organization on host associated microbial communities. And then I'm going to talk a little bit about the spatial processes that drive the emergence of or the scaling of networks across large spatial extents. So for the first sort of project, I guess, we took data that is available from many different locations in which they have sampled microbial communities within marine sponges. I don't know if you can see this, but there are some like, orange points all over the place here. And the first, as in every other microbiome study, the first thing to do is to see what are the microbes that live in each of your samples and whether there are differences, right? So it turns out in this plot, you can see the different microbial taxa on the y-axis and the different sponge species on the x-axis. And you can see that there are some patterns like, for example, there is a bunch of microbes here that are only found in these particular types of sponges or more predominantly. And, this is all nice from like a community ecology perspective, but we wanted to, try to understand better this organization. So for that, I built networks of, the microbes that live in particular hosts. And then I apply different analysis to this. In particular, we looked at the modularity of the network. And then I tried to try to understand the underpinning mechanisms behind this modular structure. I tried to relate this to the phylogeny of the hosts, right? And as it turns out, there are, there is some clustering, like for example, this orange species here. So this is a phylogenetic tree of the host species in the system. And the colors and these names are colored by the module that they belong to in that network. And the interesting thing is that there are some sort of like pieces of the phylogenetic tree that, according to the network structure, are more similar to each other based on the microbial community, which is like a strong indication that there is a co-diversification or at least phylogenetic pattern between host and microbes. And then we went on further to try to understand not only the mechanistic basis for this modular structure, but also the functional the functional meaning of this network structure. And as it turns out, this is a heat map in which we can see on the x-axis the sponge species, so the host species, and they are highlight. So in these big squares, I highlight the modules of the network for which the functions, which are on the y-axis, are significantly different across modules, right? And which this means is that the modular structure of the network actually reflects very well the different functions that these microbes perform for the host. So yeah, basically the whole what we showed here in this couple of papers is basically that that network structure can help you understand the evolutionary origins of these associations and also the functioning of the systems. So changing gears a little bit, another one of my interests is on how the structure of networks of ecological interactions, in this particular case, food webs, changes or scales across different spatial extents. And for that, I worked with a colleague of mine, Nuria Galeana, and we basically developed a a theoretical framework based on 3 different paradigms to think about spatial processes in ecological communities. One which is mostly like a null model, in which we select species based on the species area relationship. Probably I should explain this a little bit for those of you not familiar with this. In ecology, there is like this very general pattern that over the decades has become like a rule, a fundamental rule in ecology, which is known as the species area relationship, in which basically area is related canonically to the number of species that you are expected to find in that like fraction of area, basically, or extent of like size of area. And this has been observed in many ecological systems all over the world and in different like biomes and stuff. So our aim was basically to understand theoretically how this network relationship can translate into scaling of networks, not only biodiversity. The second sort of model was based on basically the trade-off between colonization and extinction in islands of increasing size. So another sort of classical result in ecology is which is known as the theory of island biogeography. Basically, it states that you can predict the number of species that you have in an island based on a trade-off between colonizations and extinctions. And we wanted to understand how these underlying processes of richness also affects the structure of the network. And then finally, we added a little bit more complexity into this, and we created a meta-community framework in which different localities were connected in a network of patches, let's say, where species live. And within each of these patches, we had networks of interactions. So you can think of this as a network of networks, and then dispersal dynamics are going on between the different localities. And in this way, by aggregating these islands, we can generate artificially like a gradient of area sizes and quantify again the properties of these networks in this across spatial extents, basically. So what you do when you look at this thing is basically you evaluate different structural properties of your network across different area sizes. In our case, there's some interesting ones here, although we explored many more, is, for example, the average link per species. And we were able to provide some predictions on how we think that this scaling will play out based on the sort of spatial mechanisms that are going on. So ecosystems in which different spatial mechanisms might be more important than others are expected to change in links per species, for example, in different ways, right? Which is sort of important if you are interested in conserving either, islands or, more continental biomes or ecosystems. Another thing that also changes differentially is the in degree or the number of prey that species have, and also the fraction of basal resources. So after having these sort of theoretical expectations, let's say we went on and took data from species distributions and the interaction, the food webs between these species across from local to very large spatial extents, in particular the vertebrate network across Europe and all of its biogeographical regions. And we were trying to see how the shape of this network area relationship looks like in these ecosystems, and obviously, check out our expectations, or maybe they will break down. So yeah, we found very similar patterns to what the models suggest. The interesting thing is that across the different bioregions, we found many different shapes of these relationships. This sort of suggests to us that across different sort of ecosystem types, there might be different mechanisms or spatial dynamics going on affecting this network. So for example, here, if you look at the number of links, there are ecosystems like, for example, I think this is the Alpine, in which, the number of links just grows on and on without like seemingly saturating. Whereas in other ecosystems, like for example, boreal, there seems to be like a limit to biodiversity. And this sort of suggests some trade-off, between dispersal ability or, potential of local extinctions and so on. So yeah, that part of the talk was mostly about the assembly mechanisms and how we are trying to study both ecological and evolutionary processes that can be giving rise to the patterns that we observe. The other thing I am interested in is in using this or models like this to understand the effects that different aspects of global change might have on biodiversity, what we usually call community disassembly. So I'm going to talk a little bit about how I have looked at invasions in complex food webs. For this particular project, basically, I adopted a more sort of a network dynamical approach in which the dynamics of the nodes in this network, which are representing species connected by many links, are simulated using these two equations. So the basal species at the bottom basically grow according to a saturating function, which is sort of roofed or sealed by carrying capacity. And then they lose biomass to their consumers. And then all of the consumers, regarding of their trophic position, they grow when they eat from something else. They lose biomass when they get eaten, and there is also like an intrinsic death rate. And the functional response in the the shape, or the shape of the function that that models the transfer of biomass from prey to consumer, but basically follows this holding type between 2 and 3 functional response, in which the attack rate interacts with the biomass of the prey, and then there is a handling time down here to avoid like overconsumption of resources. So once I have a framework like this set up, I can simulate. This is all numerical. There is no sort of analytical solutions to this. It's just a numerical approximation in which I can simulate dynamics until, for example, time step 2000, whatever number, this is arbitrary. The bioenergetic dynamics are simulated through this time, and at this point, I introduce an invasive species, which is highlighted here as the green node, and then we let the whole model to run again for further time steps, and we obtain a less complex network with less species, and we evaluate these networks using a bunch of network properties like these ones: connectivity, links per species, mid-food chain length, etc., to see how the network changed as a product of the invasion, right? And then we can also quantify, how robust is that network to the invasion based on these structural properties, and we found that connectance actually increases the likelihood of invasion. So more connected networks are more easily invadable. Similarly, networks with higher food change are also more easily invadable, but networks with strong modularity are more robust to this invasion, which is kind of nice because it shows that, when you have like different parts of your ecosystem that are more or less independent, then it's harder for a new species to get in. And then we also looked at the consequences of the invasion for stability. And interestingly, when you look at two different types of stability, community biomass, which is the aggregation of all the biomasses into one single signal, and you analyze population variability, versus population biomass, which is the average of the variability across all of the populations in the system. Basically, the thing sort of switches, right? So the fraction of stable networks for less connected systems is higher when you look at the total community level, but when you look at per population, they are less stable. So again, there is some interesting, I guess, take-home messages here, which suggest that depending on whether you look at the high level all community composition, or if you look at the population level, then stability changes when you have an invasion. So then trying to thinking about ways in which we can study different things or different stressors happening at the same time. A friend of mine in France and I came up with this idea of like using the same sort of framework, but adding some sort of temperature dependency of the of different vital rates to analyze how warming can sort of interact with invasions, right? And basically, we did the exact same thing as before, except that all of these parameters, which are like relevant vital rates that have been quantified in the field and in mesocosms as changing predictably with warming, and with a specific function, basically, how basically varying these parameters affects the outcome of the invasion. And in particular, we varied the intrinsic growth rate of basal resources, their carrying capacity, the metabolic or death rate of consumers, the attack rate of the strength basically of the interactions in the networks, and the handling time of a species. And as I said, all of these things have been observed. So the parameters for this Arrhenius type temperature scaling have been quantified for different systems in the fields. And these are the numbers that go into these parameters here, basically. So it's not, what I'm trying to say with this is that we just didn't make up all of these parameters, but they have specific values that have real meaning in real ecosystems, basically. So when we did this, basically, we repeated the simulations that I explained before across different temperatures from 0 to 40 degrees Celsius. And we observed, this sort of decreasing relationship of invasion success measured as the fraction of invaded networks. So we replicated the same simulations over and over again, and we quantified how many of those were actually invaded. And it looks like we should expect that, relatively speaking, colder communities should be more easily invadable. But the interesting thing about this part of the network is that these communities, because they are warmer, they became less complex before they got invaded, which means that, it's not necessarily a good thing that to be in a warmer environment, even though this suggests so. Then looking at the fraction of species loss, so these plots would be basically a way of quantifying the negative effect of the invasion on the network. So this is the fraction of species lost on the y-axis, and then the fraction of links lost. And you see that as temperature increases, the actual fraction of loss is way higher, right? So as I was saying before, the thing here is that temperature interacts with invasions in such a way that when you invade, even though it might be harder to invade warmer communities, the consequences are more devastating for the system. Then we also looked at other network properties, like for example mid-food chain length, and in this plot the different colors of the dots are different temperatures. So you can see here that going back to the fact that colder communities are not necessarily worse off, they in general display higher food chain length, which is sometimes sort of related to a healthier ecosystem. So colder communities are still able to preserve relatively high mid-food chain lengths, whereas warmer communities become much less less complex. So cool. So yeah, another aspect of global change I am very interested in is habitat loss, and how the loss of space in general, not only because of habitat loss, but mostly because of it, how that changes not only species richness, which is the traditional way in which we tend to quantify these effects, but more importantly, how the interactions actually are affected. Because sometimes, I mean, the interesting thing about this is that sometimes because of habitat loss, you don't necessarily lose species. So if you're only looking at your system based on alpha diversity, you might think, we can lose habitat, it's fine, it doesn't matter, until, you start losing species. But in reality, there are interactions that are being lost way before this happens. And I am very interested in understanding not only when this happens, but what are the consequences of this, right? And for this, I used like an individual-based approximation in which individuals live in this sort of artificial cellular automata type of world, which is a gridded artificial world like this picture on the left. And here I have highlighted two sample trajectories of these artificial individuals. The sort of light gray shaded area is the observable world of each of the individual in the middle. So they can only interact or observe the immediate vicinity, and then they can move randomly across the landscape. And then when these two individuals here, the black and the dark gray, after having traversed this sort of path, they meet each other here in the middle. They interact based on some rules. And these rules come from the network of species interactions, right? So in this particular case, we had 6 different types of species. And if you are a mutualistic plant and you find the pollinator that pollinates you, then both of you exchange some benefit. If you are a top predator and you find a prey, you eat it, and then that individual is removed from the world, and so on. And we wanted to understand what were the consequences of these local dynamics for the emergence of large-scale patterns. So to perform some habitat loss experiments into this world, we basically removed chunks of habitat. Like, for example, let's say here that the green parts are the habitats we preserved, and we removed, all the red habitat, and just to see what happened. And the results basically suggest, so in these plots, what we have on the on the x-axis in the top row is the fraction of habitat that was lost. And on the x-axis on the left, we have the interaction strength. And on the right, we have the coefficient of variation, which is a measure of stability of the system, the same one I was using for the invasions before. And the three columns here represent three different types of habitat loss that we simulated or that we studied. The random loss, which is we lose cells randomly across the entire landscape. Intermediate is that there are patches of habitat that are being lost all over the place. And the contiguous scenario is in which you lose an entire chunk of habitat. And the interesting thing is that when you move from a random loss all the way to a contiguous, this can be sort of, for those of you familiar with the current debate in habitat loss, whether it's better to preserve small patches or, like large areas, the habitat spare versus habitat, no, how is it called? Well, anyway, I forgot, but yeah, you see the picture. So there is a trade-off between whether it's better to lose habitat in the same place, big chunks, or the same area but distributed across the landscape. And here we can see that for random loss of habitat, as you start increasing habitat, the interaction strength between the species decreases. And also the variability of the population. So in some sense, losing habitat randomly decreases the variability of the population, which we could be considered good or not. Whereas in a more contiguous case, as you start increasing the habitat loss on the x-axis, then the interaction strength increases. And this is accompanied by an increase in the population variability. So they are less stable. And similar here, we are also looking at pretty much the same thing, but this time we are plotting interaction strength with population variability, just to get a more mechanistic understanding of this. And basically, this corroborates the fact that, for large fractions of habitat loss, which are the red points here, you have smaller interaction strength and smaller variability. And on the contiguous case, you have large interaction strengths, which mean larger variability of the populations. So this is an interesting way of looking at habitat loss, because then you can go on and ask what is happening at the individual level that gives rise to this. And basically, what we are suggesting in this paper is that basically the problem or the driver of these patterns is the reduced ability of the individuals to move in a landscape that is more fragmented, right? So these are three example trajectories for the same simulation time. And you can see here how the trajectory in a random scenario is much more contained in a very small space of a single individual versus the trajectory of this individual in a contiguous habitat loss case, in which it's more able to explore larger parts of the landscape. So that at the same time, there is a compression of populations, but predators are able to move more. So they are able to inflict like a stronger interaction strength to their prey. So that this seems to be the mechanisms behind these sort of responses. Cool. So now I'm going to switch gears again a little bit to show a couple of examples in which I have tried to use techniques like this to tackle conservation issues. The first one is how we can come up with more clever strategies to manage invasive species. And this was some work that I did when I was in Australia a few years ago, in which in Australia they have a massive problem with many different terrestrial invasive species, like the feral cats, the rabbits, and some people consider the dingo as an invasive species. Some others, do not. But anyway, and the focus of the study is what happens when you try to manage these species, such as, for example, the rabbit, on small marsupials, which, ever since, hundreds of years ago, Australia has seen massive declines in their populations and the extinction of many of these species because of the introduced ones. And they are always trying to find ways of, making these management things more clever. So for that, basically what we did was we took, this time it was more like a different type of equation approach rather than continuous. And the interesting thing about these systems is that we had specific values for the parameters of specific things. So for example, plant biomass, which was the basal resource, grows using grounded on this specific functional form. And similarly for consumers, we had, based on empirical data, we could infer the exact of shape in which they respond to their prey. So it's not very important to understand details here, but this is just to show the type of approach. And basically we use those equations to simulate the dynamics of this network. And then once we had a system that that showed the similar behavior to the actual one, we removed this little guy here to see what happened. And after removing that, we removed rabbits at different fractions. So like 10% of the population, 20%, 30%, all the way to 90%. And we quantified the response of the system based on the abundance of the little marsupial species. And what we found of interest here is that you have there is the removal of the invasive species it's all nice up until certain threshold, after which you are actually doing more damage to the system than what you are benefiting it. And the reason for that is that there are all sort of like percolating effects through all of these other species. And in particular, we observe what it's called the Mesopractor release effect, in which not the Mesopractor release effect, the hyperpredation effect, which happens when you remove the prey of a particular consumer, it will start relying on the other prey. So even though this was sort of intuitive to follow, the full consequences of this were not really revealed until we performed these experiments and saw what happened. And then, well, we did this for different sort of scenarios of control, and we also evaluated the behavior after the actual removal of the species to see what how dynamics would play out through time. And this is what we found. The last thing I want to talk about is a little example on how how to use these networks and also individual-based approaches to, in this case, try to manage the spread of diseases in wildlife population. In particular, we focused on the Tasmanian devil facial tumor disease. So this is actually a cancer, a tumor that grows in their face, but it's transmissible. So they pass it on to each other when they bite their faces. And it's like a very big problem in Tasmania, basically. So one of my MRes students last year, she took basically the habitat availability information for Tasmanian devils across the entire island. And based on this, she created a network of local patches with some connectivity based on the distance between the different habitat patches. And within each of these local populations, we had individuals that interacted with each other based on basically fighting with each other and with some probability of the transmission of this disease to one another. And we evaluated different dispersal scenarios and different transmission scenarios to basically assess the effect of the animals moving across the landscape and the transmission rate of the disease in the consequences of that for the prevalence of disease across the regional scale, so across the entire landscape, basically. And it turns out that, you know, for very low levels of transmission, basically the disease is expected to die out, even at large levels of dispersal, which are here shown in the x-axis. The y-axis shows the proportion of populations that the disease has reached. And the interesting part of the parameter landscape, I guess, is between 0.3 and 0.4 transmission probability, because here the ability of the disease to reach different parts of the population of the landscape basically is very much a function of not only the dispersal but also the contact distance between the individuals. So how basically a measure of how likely it is that they will fight with one another. And we found that dispersal is very important for like the first stages of the disease. So if dispersal increases a little bit, there is a big difference. But then after that, you know, the strongest determinant is how likely it is that those individuals will fight. And yeah, I guess that the interesting thing is that, you know, or the take-home message here is that if we want to prevent the disease spread, we need to find ways of, you know, keeping the individuals so sort of sparse throughout the landscape. This is basically the result that she got from our model, which I just wanted to show this nice animation because it really shows the disease wave front, how it, you know, from the emergence of the disease back in 1996, it basically wiped through Tasmania. This is an actual outcome of our simulation, but it replicates really well what was observed in the field. So, and then once we had that framework in which we understood better the interplay between dispersal and disease transmission, we could play a little bit with it by designing basically strategies to manage the landscape based on the topology of the network. So we use 3 different management strategies that we call bitwinness, degree, and random, which is basically the removal of nodes in the metapopulation based on their bitwinness score, based on their degree, so how many other population it is connected to, or just removing randomly. And even though for the total metapopulation size or the number of individuals at the landscape level, there are not many differences, for the proportions of patches infected, it is very important whether you manage your populations randomly or if you use a more clever strategy like looking at or quantifying the bitwinness of those local patches. And if you do that, if you are able to quantify, you know, what are the patches or the local populations through which devils sort of transit when they move throughout the landscape, then you have higher chances of and you manage those patches, you have higher chances of decreasing the infection at the large scales. Genetic variability didn't change much across landscapes, so that was, I guess, an interesting result, even though we expected some differences, but there were none. So yeah, I guess that's pretty much it from me. I hope you enjoyed what I had to say today. I mean, I'm not gonna read these conclusions here because it's you know, just for us to look at, but basically what I'm trying to get at is that you know, we can understand a great deal of the ecological and also evolutionary mechanisms of assembly in complex communities by using network approaches. I am very interested in that. I am also very interested in using like network modeling approaches to understand the scaling of biodiversity, and also to understand the effects of stressors in natural communities. And I am always interested in trying to apply this knowledge to conservation problems. Thank you very much.
Thank you, Miguel. So it's time for comments or for questions. If you have some questions, just unmute yourself and you can ask to the speaker the question. To start with, I have one myself. Is in the example of the assembly in which you take into account the biogeography, the second example you have shown, I think. Could you give an idea of the type of models you were using? Is it
sorry, I don't know why you lost my screen, right?
Yeah, you have to share again.
Yeah. So you're talking about this?
Yeah. So which, I mean, you had some results for the scale. Okay, this kind of thing. So what is the model behind?
So yeah, for each different color line here is one of these different 3 models. So we had 3 models, right? So the first one is kind of a null expectation, which is what would happen if you just, you have a network, because we start with the knowledge of the entire network. So we know the network, right? Mm-hmm. And then in the null model, we choose a certain number of species randomly based on the network area relationship. So basically, in this plot, that null sort of expectation model is the blue line, and here, if we have, I don't know, an area, this area is relative because for comparison, we standardize it between 0 and 1. But let's say that this area is, I don't know, 100 hectares or whatever, right? So this is our first area. So we go to the network area relationship and we say, well, right, for this area, we are supposed to have 20 species. When we do that, then we go to the network and we choose 20 species in that network randomly, and we quantify those properties. And then that would be a point here. So the links per species in a network with 20 species in which those species were chosen randomly is this number, whatever, 6, right? Then then we have 2 more models. So the green line here is the second model, which is the island biogeography model. So in this case, similarly, it adds a little bit more of complexity because it takes into account the trophic constraints. So in the first model, we don't care if the species have predators or not, like it's just a random collection of species. In the second model, we select our number of species based on the colonization extinction trade-off, and then but we ensure that those species actually have prey, so that you cannot choose a species without a resource. So, and this is what happens when you add that constraint. This is what happens in the green line, basically. So we go there, we say these are for this area, this is our colonization versus extinction trade-off. And that number gives us this number of species and this network, and this is how we quantify this network. And then for the last scenario, which is the meta-community framework, is this one here. Basically, we have, I think we did it with 75 patches. So we have 75 patches. In each of these patches, we inoculate with some species at the beginning, and then we allow them to disperse between patches. And then we go to this plot here, and the reason why there are two colors is because we tried with two dispersal rates. So the red one was a lower dispersal rate, and the yellow one is what happens when you have a lot of dispersal in your landscape. That's also why, you know, the number of species starts very high in this plot, because when you have a lot of dispersal, basically you homogenize the landscape. And here the model works like this. So we simulate the dispersal of species between patches for some number of iterations until you know, the whole landscape remains stable. So there are no more species that are able to colonize new patches. So everyone who was able to colonize everything, that's it basically. And then for area, for example, 25% in the, or 50%, which is out of 75 patches, that is 37.5. So we aggregated those patches and constructed one full network from all of these networks of 30-something patches. And this is the value of links per species at that level. So we have 9 or whatever number of links per species. And that's how we came up with all of this, with all of these lines, which basically gave us expectations of what should we expect if our system is more of an insular system, like for example, a series of islands, which would be the model in the middle, or if it's more like a terrestrial landscape in which species can move more or less freely between different patches, which would be more, you know, like the second type of thing. Sorry, the third model, let's say. And these are, yeah, basically the theoretical expectations for those things.
Good. So, more questions? More comments?
Yeah, Miguel, I have a couple of questions, but maybe just one for now. So it's quite broad, your interest, right? From spatial scaling of biodiversity, invasions, pathogens, right? The dynamic in space. So it's quite broad in general, right? Along many
Yeah.
But I have a question about the.
Sorry, just to say something quickly. To make things even worse, I'm now trying to add evolution into the whole thing, eco-evolutionary dynamics. So it's gonna get even more interesting.
Yeah, that's great. I have a question about maybe your vision about the trends, the future of modeling, null models, but also mechanistic modeling, right, in these complex systems. So if in most of your approaches you have interaction traits or interactions, but also environments that usually are modeled using abiotic traits. So you have this dilemma, right? You have biotic and abiotic traits. And if you take a look at the literature in detail, there is a kind of, you can correct me here if I am wrong, but I feel there is a strong gap in understanding trait architecture for spatial scaling, for example, right? Accounting for biotic and abiotic traits. What do you think about this? What do you think about the extensions, right, of mechanistic modelings of biodiversity, including different traits? Or how do you see this? How?
Yeah, no, I totally agree with that. I think that, well, the models I presented here obviously do not account for abiotic heterogeneity at all. You know, like, well, we tried to do that a little bit with the European biogeographical regions precisely because of that, because we suspect that different environments shape this scaling differently, and that's what we were trying to get at here. But I think that incorporating this, you know, like these aspects into modeling frameworks will require like an added level of complexity, and I will explain why. So when you have like a network of the interactions, you know that the species trait that you are interested in is the attack rate, for example, or that thing. But then the environment sort of at the same time, it's affecting like a different layer of the interaction, which is it's affecting the vital rates or the different things of each species differently, right? So in here I took the, let's say, general approach of assuming that, for example, in the case of warming, all of the species are responding at in the same way. But I think that that the challenge would be to add traits that somehow interact with the interactions and that encapsulate this environmental information you're talking about, to be able to understand how environmental heterogeneity not only acting on the biology or demography of the species, but also on the interaction as a different thing, changes our view of things. So yeah, it's it's a very interesting challenge. I don't know if it would be feasible to tackle, because it's sort of hard, but it's something. I mean, in the ecoevolutionary stuff that I am trying to do now, I think, because in my framework, the only thing that is evolving is the interactions, and I am thinking all the time of ways in which we can evolve other things that adapt to different parts of the abiotic multidimensional niche of the species, and how that would not only affect the range of the species, the spatial distribution, which obviously will have consequences for the realized interactions, but also will affect the actual interaction per se, right? So that those are the two things. Interactions can be affected by what the species is doing on its own, based on the environment, but it can also be affected as the interaction per se, if you see what I mean. So yeah, I don't know. I don't know how to solve that. Yeah.
Thank you, Miguel.
Any more question?
I have a question regarding timescales. I mean, you have tackled different problems, we discussed different timescales, and now you are even talking about this evolution thing that is, of course, at much longer timescales. So the point is, for instance, the propagation of that cancer that probably took several years to propagate, because biting one animal to the other. So what are the typical timescales that you consider relevant for the different problems, and how the different timescales play important in the dynamics? Do you really need to model short timescales for the long ones? I mean, what sort of ingredients you are going to disregard when going to longer timescales?
I guess that's a hard question. I mean, I have never, all of my models or all of the stuff I presented here is usually relevant for like a specific time scale. So I have never found myself in the necessity of like coupling different time scales.
Okay.
Which is, I guess that.
But for instance, now you are talking about this evolution of the links by itself, or the interaction, or how you.
Yeah.
This probably occurs on a much longer time scale than the normal interaction by itself. So naturally you have to have at least two time scales, one for the day-to-day and the other one for the changes over, I don't know.
Yeah. So what I do in the stuff that I am doing now, which I didn't show here, basically is that the ecological time scale happens, I run the dynamics using equations like the ODEs I showed here. And then when I achieve stability, this is a very simplistic way, and it might be wrong, but when I find the stability of the system at that point, is when I introduce an evolutionary change. So basically,
Is what we call an adiabatic evolution.
Sorry?
Is what we would call an adiabatic evolution.
Oh, okay, I wasn't aware.
You allow to the system to reach some equilibrium, and then you change some parameters, allow the system to reach equilibrium, and so on.
Yeah, yeah. I wasn't aware of the terminology, sorry, but yeah, that is exactly, yeah.
Also thermodynamics. It's the change that is done in such a way that you always maintain the system at equilibrium. So the parameters are changed very, very slowly.
Yeah. Yeah, but of course you can think of ways in which you can actually incorporate the evolutionary part into the dynamic equation by setting a very small mutation rate. So so because the way in which I change interactions or I create new ones is based on some probability. So if the dynamics are simulated for some time, and that parameter is very small, then you kind of expect that the stuff that is happening ecologically, it's going to last for much longer than when the next evolutionary thing happens, basically. So yeah, I could see that. I am not doing it like that now, but I could see how something like that could work. Yeah.
Well,
Yeah.
Thank you.
So
Yeah,
there's
no,
now some fast question or fast answer. Do you have some short question? If not, then let's thank Miguel again. And okay, let us close the session. Thank you to all. And okay, and Miguel, we will remain in touch. We have to talk about some things. Okay.
Cool. Thank you.
Okay. Goodbye.
Yeah.
Bye, everyone.
Ciao, ciao.